All right, in section 3, we're covering mutation. So section 2 is replication, and mutation is the process of errors getting incorporated into DNA replication. So we've talked at length about how good DNA is at replicating itself, but sometimes mistakes happen, and so we call those mistakes mutation. And so we want to understand what causes those mistakes, what the consequences of those mistakes are, and what are the various ways that we can categorize those types of mistakes. So, we think about DNA in terms of being our double helix, and then we also think about it in terms of our base pairing. So in order to think about the causes and consequences of mutation, we look at it from both points of view. So one of the first causes of mutation is radiation. And so we're going to broadly categorize radiation into two categories: ultraviolet radiation and ionizing radiation. So ultraviolet radiation, right, is happens when we expose ourselves to the sun, and that ultraviolet radiation pierces our skin and it makes it into our cells and it hits our DNA. And ultraviolet radiation basically causes changes to our DNA that makes it such that replication is less effective or ineffective. In essence, places where we have adjacent pyrimidines, say example a thymine and a thymine, or a cytosine and a cytosine, we have the opportunity to lose the hydrogen bonds that we have across from those pyrimidines, and we have a dimer that forms with those adjacent pyrimidines. So ultraviolet radiation causes that to happen, which creates problems for DNA replication, which we'll talk about in a minute. Ionizing radiation, like X-ray radiation, basically comes in and provides enough energy to break the phosphodiester bonds that link our sugar phosphate backbone. So our ionizing radiation comes in and just breaks our DNA, right? And so when you break a chromosome, a linear chromosome, then part of it is going to be lost unless you can find a way to fix that very quickly. And then we have all kinds of things that we call mutagens. And a mutagen is just an environmental circumstance or an environmental compound that increases the likelihood of a mistake happening during DNA replication. And there are all kinds of ways that mutagens operate, but one of the easiest ways to think about it is that mutagens will change how our bases interact with each other or how they behave. So as an example, they might take this adenine and chemically modify it such that it behaves like a thymine, as an example. Happens in all kinds of ways, but that's just a simplified way to think about it. It changes how those bases behave. And what that means is when we replicate this piece of DNA, the next time around, this bottom strand will behave normally and it'll get copied with an A, but this top strand, this A, right, it is an adenine, so it should get copied with a thymine, but it's not going to get copied with a thymine, it's going to get copied with another adenine. So that creates a mispairing. We're going to have an adenine paired with an adenine. And then one of the most common ways that we can mutate our DNA happens as a product of normal processes inside of our cell. So at the end of the electron transport chain, we're pumping hydrogen ions through ATP synthase, we're making ATP, and then we're catching those hydrogen ions with a single oxygen, and we're making water. But that process is imperfect, and some of those O1s, some of those oxygen-free radicals escape, and they're highly reactive, right? They want to react with anything that they can react with, and one of the things that they'll react with are the nucleotides inside of our cells. And so they, in essence, what they're going to do is they're going to damage those nucleotides such that they don't behave normally. So in the same way that that mutagen changed the behavior of that adenine, oxidative damage is going to do the same thing. All right, so that's kind of a nutshell of how, our DNA gets changed or damaged. But then, what's the consequence of that after DNA replication? And so what we say is, we have big picture, we have chromosomal mutations, and chromosomal mutations are where we have ionizing radiation that basically shreds a chromosome. So anything that is large scale, where we lose a lot of DNA, we call those chromosomal mutations. But small scale mutations we call point mutations. And so these, a deletion, if we lose a lot, it could be a chromosomal mutation, or it could be a point mutation if it happens at a smaller scale. So in terms of talking about our mutations from above, ultraviolet mutation can create those dimers between adjacent pyrimidines, such that when we copy our DNA, they create a deletion. We lack the ability to add a hydrogen bond to those. So in this particular example, my bottom strand can be copied as normal, but my top strand cannot be copied because I can't form hydrogen bonds because of the ultraviolet damage to those. And so that's going to give me a deletion. I'm going to lose those two cytosines. Similarly, I can also get an insertion. And so one of the things that I didn't mention up here is viruses and transposable elements. And so anytime I have a transposable element, it takes a piece of DNA from somewhere in my chromosome, and it picks it up and it puts it somewhere else. So that's a transposable element. So transposable elements are pieces of selfish DNA. They make copies of themselves and they insert themselves somewhere else. And when that happens inside of a cell, we just call it a transposable element. But when it happens between cells or even between organisms, then we call it a virus. A virus is just a transposable element that has gained the ability to move outside of its host cell and into other cells, even into other organisms. And so anytime that you have a virus or a transposable element, you have the opportunity for a deletion and an insertion, right? A piece of DNA that's cut from one place or copied from one place and inserted somewhere else. When we're talking about our mutagens or our oxidative damage, we're talking about point mutations, things that are affecting small numbers of base pairs, and one base pair in particular. And so when we're changing the behavioral properties of a base, then we're making it behave like something else. And so anytime that we have a purine that is behaving like another purine, we call that a transition. And anytime that we have a pyrimidine behaving like a purine, or vice versa, then we call that a transversion. So our purines are adenine and guanine, and our pyrimidines are cytosine and thymine, as we reviewed in week 1. So if I take an adenine and I mutate it such that it behaves like a guanine, that's a point mutation, but I've substituted a purine for a purine, and so I have a transition. But if I take this guanine and I mutate it such that it behaves like a cytosine, now I have a purine that is functionally a pyrimidine. So I have a trans version. So DNA replication is very efficient. The structure of DNA allows for very, very precise replication, but mistakes can be made for some of the reasons that we just discussed. And so in a lot of those situations, we can fix those mutations before they can have consequences, downstream consequences for the cell or the organism. And one of the things that we talked about is ionizing radiation, which creates double-strand breaks in our DNA. And one of the things that we can do to fix that double-strand break is called recombination or homologous recombination. And so for diploid organisms, right, like humans, we have 2 sets of chromosomes. So if we have one chromosome that experiences this double-strand break, we have, in theory, we have another chromosome that's identical to it, and so we can line those 2 chromosomes up together, and I can use the information from this homologous chromosome to fix its homologous partner. So we call that homologous recombination. But sometimes in that situation where we had that double-strand break, we don't have a homologous chromosome available for whatever reason. And in that case, we can use non-homologous end joining. We can just, if we catch it quick enough, we can just glue those ends back together. Now, maybe we haven't lost any information, maybe we have, but having those chromosomes connected is better than not having them connected. And then in terms of point mutations, we can fix point mutations. So we have various ways of finding situations in which we have mispaired bases. So if we have an adenine paired with an adenine, that's going to create a physical distortion in our double helix, and right, they're going to be imperfect hydrogen bonding. So we can see that that's happened, and we can fix it. And in essence, what happens is when we find those errors, we excise the incorrect bases, and then we have DNA polymerase come back through and fix the mistake. And so there are a handful of ways that happens. Mismatch repair is one of those examples. But for most point mutations, we can find them and we can fix them. And then, so one of the things that we talked about was ultraviolet radiation, and so if we have ultraviolet radiation that comes in and creates a dimer between these adjacent pyrimidines, in theory, when those go to be replicated, they can't be replicated. So if we're trying to copy this, and we can't form hydrogen bonds because those polarities have been, we've lost those available polarities, so we can't bring in those adenines. So we can use what's called translesion polymerase. This is our lesion, and we can just go across it instead of having a deletion. And so translesion polymerase doesn't know what those are because it can't see them, because it can't form hydrogen bonds with them, but it can just add random bases. And so that's going to give us mutations, but at least in some situations, having point mutations is better than having deletions. All right, so we have mutations. We can fix some of them, but we don't fix all of them. So what are the consequences, and how do we characterize those mutations? So most mutations are going to be somatic cell mutations. They're going to happen inside the somatic cells of an organism, and they're only going to be of consequence to the cells that arise via mitosis from those mutated cells. So those are of consequence only for the organism that experiences them. But if they happen during meiosis, which we'll talk about in the next section of the cell cycle, and they happen in the germline cells, then they're going to affect downstream generations. So when we produce gametes, And during DNA replication in the M phase of meiosis, then we're producing gametes that have those mutations, and those affect future generations. So they have the ability to affect future generations. So those are the mutations that give us genetic variation, that gives evolution the tools that it needs to adapt or change over time. Another way that we can categorize mutations is either by gain of loss, or gain of function, or loss of function. So if for some reason we increase the activity of a gene, then we would call it a gain of function. But if we lose function, or if we decrease function, then we call it a loss of function mutation. And for our point mutations, right, we're changing one single nucleotide. Those mutations can be missense if we change the amino acid that a codon codes for. So anytime we change our amino acid, then we are changing the sense. We have a missense mutation. If we change an amino acid to a stop codon, then we have a nonsense mutation. And if we have a stop codon that changes to an amino acid, then we have a nonsense suppressor. And those are all things that we're going to revisit when we get to transcription, translation, and regulation. Another way to think about our mutations is whether or not they are forward and reverse. So one of the terms that we've talked about is wild type versus mutant. And the simplest example is at gene A, where capital A is wild type and little a is mutant. So we call a mutation that takes us from wild type to mutant as a forward mutation. And then we call a mutation that goes from a mutant back to a wild type a reverse mutation. And then one last way that we can think about mutations is whether or not they're functionally consequential. And so if a mutation is synonymous, that means we have a missense, but that missense does not change the protein. But if a mutation is neutral, that means that we've changed the bases, but we haven't changed the amino acid.